the structure of starch and cellulose, then we have authority to see how starch really relates to the roles. Okay? Yes, we know the roles of starch. It is a storage compound in plants. But which properties does starch possess that let starch, that allow starch perform its roles? Okay? Can you think about some few? Just think about some. Yes, starch is a storage compound. Think about some. Which roles does it possess? Okay? Which adaptations does it have that allow starch perform its function as a storage compound? So let's talk about some few. Number one, it's a large molecule. Okay? It is a polymer. So being a large molecule, it makes it relatively insoluble in water. In fact, starch is insoluble in water. It doesn't dissolve. And if you want to try, just make some cassava flour and you put it in water. Get your portion, maize flour, and you dissolve it in water. You will see it won't be able to dissolve. Okay? It will form that white PPT. So meaning starch is insoluble. And so being insoluble in water, first of all, it won't impact on the osmotic pressure inside the cell. Secondly, it won't easily diffuse out of cells. Okay? It cannot diffuse out of cells. And so because of that, then you can easily store the starch. That's why even after photosynthesis, during photosynthesis, the sugars that are formed, actually you're going to form glyceraldehyde, okay? Phosphoglyceraldehyde, that's what is going to be formed. And then the two molecules of phosphoglyceraldehyde will combine to form glucose. But now the glucose that is formed, it cannot stay within those photosynthesizing cells. It will greatly impact on the osmotic pressure of these cells. So as soon as the glucose is formed, then it will be rapidly condensed to form a starch. And so the starch, being a relatively large molecule that is insoluble in water, it won't impact on the osmotic pressure of the cell, and also it won't tend to diffuse out of the cells. And so it will be ideal for storage. Not so? That's brilliant. Number two, our starch, it is linked by weak, I can say weak, and not that weak. They are strong enough to hold the glucose molecules together, but they are weak enough to easily be broken down, to easily be hydrolyzed, to produce glucose whenever it's needed. So the starch is linked by the weak alpha 1,4 glycosidic bonds. Now that are not too strong to be broken, and they are not too weak to be overcome with ease, but they are relatively strong to keep the free glucose molecules together to form starch, but also to easily be hydrolyzed in need of the glucose, okay, which ensures starch to be an ideal carbohydrate for storage. Is that okay? In case you have any query, feel free to contact. Then, as we have said when we were discussing, we saw that the starch forms cross linkages that are internal, okay, and so as a result, these molecules of starch will end up forming these internal cross linkages. It will lead to high recoiling of the starch molecule, so that it is really coiled, okay, and so this high recoiled structure is good for storage. The sparking space is going to be limited. The storage space is going to be minimized. Why? Because it is highly coiled. But now imagine if they were just long fibers. Just long fibers. Which one would be easy for storage, and which one is hard as far as storage is concerned? Obviously the one which is highly coiled. So the one which is highly coiled takes up little space, but you pack enough than if it was just a straight chain. Okay, not so? Yes. And because because these groups, the OH groups, they do project inside also, okay, then they won't be in free space to interact with the water molecules, and so it renders starch to be insoluble in water, and so being insoluble in water, it won't impact any osmotic pressure, and so you can easily store the starch inside the cells. Is it okay? In case you have any queries, the chat box is open for everyone. Then the chains of alpha glucose units can be folded to form starch grains, which are easily stored. I talked about this. So basically, that's how starch is an ideal compound for storage. Not so? So basically, those are the adaptations of starch. Now somebody was like, what is adaptation? Adaptation means how structure relates to function. It is even here. How structure relates to function. So that means you must know the function, you must know the structure, and then you try to relate structure to function. For example, adaptations of you to survive. If you say you have eyes for seeing, that is wrong. Eye is a structure that you have. Function is seeing. Function is why do you need to see? Maybe to see danger, to see obstacles as you are moving so that you are not knocked down by cars and stuffs like that. Now that is function. But how is I relate to that function seeing is an adaptation. Just saying I have eyes for seeing, it is not enough. But how do your eyes relate to function? That's now adaptation. For example, you can say, I have large. Are you hearing? Large swollen. Are they even swollen? Protruding eyes. Now that one, it makes a lot of sense. The eyes are protruding, and they are large. So that one you are trying to describe. And then how does that relate to function? It gives you a wide field of view, wide coverage, so that you view objects in long distances and wide view. You have a wide view in a wide coverage. That's why you are focusing on the screen, but when something happens besides you, you jump. If somebody stands just the other side far, assuming you are in compound, and somebody throws a stone, you jump. Yet you are focusing on the screen. Why? Because the eyes are large and protruding. But if you just say, I have eyes for seeing, now that one is not an adaptation. So these are the adaptations of starch. Let's talk about glycogen. Glycogen, just like starch, is also made up of very many alpha glucose molecules condensed together to form an alpha 1,4 glycosidic bond. Alpha 1,4 glycosidic bond. However, now, unlike starch, for glycogen, for it has branched chains, and the branching occurs at 1,6 glycosidic bond. Between one carbon atom of one alpha glucose molecule and the 6th carbon atom of another glucose molecule. And so the bond is formed is alpha 1,6 glycosidic bond. I'm going to show you, have no worries. Now the chains in glycogen, they are not as long as that in starch. They range from 1 to 20 glucose units. Okay? And for glycogen, for it is highly branched. Highly branched. Unlike starch. Now for starch, it is made up of amylose and amylopectin. Now for amylose, it's not branched. It is this amylopectin that shows some branching. But for this one, it is highly branched. It is highly branched. You can see, you can have a look. You can have a look. So this is what we are talking about. You have normal chain of alpha glucose molecules. You can see if this is the alpha glucose. Here you have this one for glycosidic bond, another one for glycosidic bond, another one for glycosidic bond. Okay? To form one chain. But within glycogen, then you have branching. So where at this, now I seeing this carbon atom, this is this glucose molecule, this is carbon 1, this is carbon 2, this is carbon 3, carbon 4, carbon 5, and carbon 6. So there is branching between carbon 6 and carbon 1, okay, of the glucose molecule. But these normal glycosidic bonds that are not formed due to branching, they are formed between carbon 4 and carbon 1. So therefore the bond formed is going to be alpha 1,4 glycosidic bond, whereas here the bond is going to be alpha 1,6 glycosidic bond. And there is high branching, you can see. As if this is not enough. Look at this. Look at that. So that's your glycogen. Highly branched. And they are short chains. Okay? Short chains. Not as compared to starch. The chains are relatively short. But highly branched. Now think about this. Why do you need to do the branching? Why do you have to branch like this? This is a road. To reduce the parking space. To park as much as you can. Okay? In little space possible. Otherwise, if you can't, then you just store 20, 30 glucose molecules and the cells are full. And you have nothing. You have no glycogen granules. You have no granules to store the glycogen. But because of high branching, then you store enough in a little space possible. Imagine all this, if this, it is just to be linear. How long would it cover? How long? You need relatively 2 cells to store just this. But because they are highly branched, then you can easily store them in this small space. Are you seeing? So that is what branching, that's why branching is good. Any query as far as glycogen is concerned? Per saying, no query, which is good. Cellulose, we talked about it in the previous lesson, but yeah, we can also have a look at it. It is 50% in plant cell walls, and in cotton it makes up to 90, you can imagine. After all, you use cotton to do what? To make clothes. We are going to see the uses of cellulose. It is made up of roughly 10,000 beta glucose molecules that are condensed together to form long and branched chain. And very many chains lie parallel to each other to form the cross linkages that link these chains together. Okay? That form these chains together. And so the property. So now imagine if cellulose wasn't a straight chain, if it was highly coiled, how did it form structures? How did it form cell walls? Think about it. How? Like how? Seriously? Like how? But now because our cellulose forms the straight chains, the cross linkages, they are formed between two adjacent chains of cellulose, then you form the straight chain structure of cellulose. Unlike starch, whose cross linkages are formed within the same chain. You can have a look at this. This is one chain. This is another chain. Another one. And you are forming cross linkages within the chains. And so you maintain this parallel shift, this parallel arrangement of cellulose. And so when you are forming the cell walls, you easily form cell walls, as we are going to see. So the OH groups, they do project outwards from each other or from each chain. And so when the OH groups, or when the OH group of one chain, for instance, okay, approaches another OH group, this is H, okay, approaches another OH group, okay, of another chain, this is O. So because these ones, they have partial charges, and this one is partially negative, the same partially positive, and in this one partially negative. So when they approach each other, then they'll end up forming cross linkages. Okay? Cross linkages between the strands. This is one strand and this is another strand. Now there are those cross linkages formed between adjacent chains, that maintain this parallel structure, this parallel sheet of cellulose. Is it okay? Does it make sense to everyone? Okay. Okay, so it seems there are no queries. So let's talk about the structure of the cell wall. Because somebody complained that in cell biology we didn't study cell wall. And I said we shall study it. I even gave you an assignment. So this was the right time to study a cell wall, because now I wouldn't talk about cellulose. I wouldn't say that cellulose is made up of myofibrils, microfibrils, not myo, and macrofibrils that are arranged in parallel sheets. You wouldn't know what parallel strand is and how the parallel strand is formed. But here I have really authority to talk about it. I'm really authoritative to talk about it. So you can have a glimpse at this. So you get your leaf, you extract one cell, you extract a cell, okay? And then you view that cell under microscope and you extract. You just concentrate on the cell wall. This is what you see. Okay? You just see my macrofibers, like ropes, big ropes, big ropes, big ropes called macrofibers that actually do interlock within each other. So now these big ropes, these big fibers, they are the cellulose fibers. Okay? They are the cellulose fibers. That when you try to study one fiber, when you try to study one fiber, then you find this one fiber made up of very many other small fibers. You can see this one, the microfibril. They are very many, very many. And when you try to study the one microfiber, then you see very many strands of cellulose. And so that's how a cell wall is formed. And then the rest you just need to have gums and binders to bind these strands together. Okay? To bind these strands together so that you can now form an overall structure, of a cell wall. Okay? Yes. So basically it is as simple as that. Not so? So basically I hope right now you have a clear view of a cell wall. You now know what a cell wall is. Okay? Now the rest is describing. So it is made up of very many chains of beta glucose molecules. These ones. These many chains of beta glucose molecules, they do associate in groups of 60 to 70 to form what we call the microfibrils. These ones. So that's what I was telling you. Okay? So to form very many fibers of to form microfibers. Are you seeing? Are you seeing? Are you seeing? Now these microfibers, okay, they will associate also very many, but they must have a binder. Okay, now you have lignin. Now lignin, they are like fibers, small fibers that will run through these very many strands, okay, to form like a binder. It's like when you have very many like ropes, and then you pass through one single rope to bind them, to hold these ropes together. So now the lignin is the one which will bind, which will pass through these very many, okay, microfibrils to bind them together. Now the strands of microfibrils, these ones, they also associate in groups, very many groups, to form what we call macrofibers. Like I told you, macro is big. Okay, it's like you begin with one strand, and the one strand is of glucose molecule. Okay, the one strand of glucose molecules. So these one strands, they associate together 60 to 70 to form one fiber, and the fiber is a microfiber because it is small. Then these microfibers, many of them, they will also associate together. They will also associate together. They will also associate together, okay, to form one large strand. Now this is another strand, and this large strand, it's a macro fiber. Okay. Now it doesn't stop there. Now you have binders, and I think in this case they are pectins and hemicellulose. They are the gums, they are the binders that will now bind these macrofibers, these large strands. Okay? Then another large strand somewhere. Large. Okay? Large strand somewhere. Okay? To form now one big fiber, which is a cellulose fiber. So now this is our cellulose fiber. Okay? These are the cellulose fibers. But when you go deep, you form a macrofiber. Then you go deep macrofibers, you find their microfibers, and then you go deep microfibers, you find their cellulose strands. And actually the same arrangement occurs in your muscle cells. Okay? Muscle cells. That's how also they are arranged. So you can have a glimpse, you can go and check in the muscle cells and enjoy. So that's basically the structure of a cellulose cell wall. If they say describe our cell wall, that's what you describe. Then the microfibers and macrofibers are held together by hemicellulose that gives them great tensile strength. And then the straight chain molecules which project both sides of the chain at alternative positions. I mean these ones, these cellulose fibers, in alternating heads and tails. These cellulose fibers, they are straight chains, okay, that project from both sides in an alternating fiber. So if this fiber runs in this direction, then this fiber is going to run from this direction, then this one runs in this direction, and so on and so on, until when you form a cell wall. And then this cell wall is going to surround the overall cell. I hope that one is now okay. Okay? So basically that is the structure of cellulose and a cell wall. So after studying the structure of cellulose, and after studying the cell wall, can't you really compare the two? Can't you really relate the structure of cellulose to the structure of cell wall? Can't you really give adaptations of cellulose to its functions? Yes, you can, and good. You've brainstormed about some. Okay, let's see if you can. Let's see. Let's mark ourselves. So number one, nobody talked about this. The cellulose, it is a polymer of straight chains of beta glucose units. Nobody talked about it. You assume to be obvious, but all of you are talking about cross linkages. But do you know what creates the cross linkages? It is because of the chains of beta glucose units. Now, because they are beta glucose units, when condensing, the second glucose unit must somersault. It must rotate at 180 degrees. So the process of rotating makes the OH bonds to appear at different sides, hence making cross linkages with adjacent cellulose fibers. Okay? And you know that the advantage of forming those adjacent fibers. You would have parallel sheets. You would form parallel strands. Okay? Which will add up to form a microfibril, and then microfibers add up to form macro, and macro add up to form cellulose fibers. And hence you have a cellulose cell wall. But it all begins with beta glucose units. And number two, the beta glucose units are joined together by strong beta 1,4 glycosidic bonds, which cannot easily be hydrolyzed. Can you mute? Anita, can you mute? Which cannot be easily hydrolyzed. And so what does that imply? It implies that it will form a stable strand of cellulose fibers that is strong enough to form a cell wall. And you know the functions of a cell wall. You prevent further expansion of the tonoplast together with the cell membrane. But if you are weak, then it will keep expanding until when the cells, boom, burst. But the cell wall prevents bursting. So you must be strong enough to prevent bursting. Otherwise, boom, your cells will burst. And so nobody talked about this. That is a checkmate. Then another one. Now all of you talked about this. And this one comes from having cross linkages between adjacent cellulose fibers. But it's really good. It's really good. So the cellulose fibers, the macrofibrils and the microfibrils, and the cellulose strands, they are all parallel. Okay? They are all parallel, which creates the cellulose to be fully permeable to water, because the fibers will be parallel to each other. The cellulose fibers within a cell wall, they will be parallel to each other, which creates space within the cellulose cell wall. You can see. And so these spaces, they are the ones that will fully allow movement. And remember, these strands, they run in opposite, side to side, in opposite directions. So they will fully allow materials to cross through these spaces. making a cell wall to be fully permeable to water molecules and dissolved solids. And so this is good as far as transport is concerned. Transport of water before it reaches the xylem is concerned. This is really good. Okay? You study in transport, you study what we call the apoplast. Have you ever heard of this anywhere? The apoplast pathway. And you see that almost 90% of water transported in the cortex cells, 90% of food transported in the mesophyll cells, it is transported through the apoplast. Why apoplast? Because the cell wall is fully permeable to water and dissolved solids. And so, what does that imply? It implies that materials will fully move across. We talked about this, which was brilliant. I've talked about this. The glycosidic bonds holding the beta glucose in cellulose are strong. Okay? And they are only broken in presence of an enzyme responsible for that. And which enzyme? Cellulase enzyme. Okay? This is supposed to be A. I think it was just a typing error. The enzyme cellulase. And so, since this enzyme is able to break down that bond, then animals that feed on cellulose are able to obtain the glucose and respire to produce the energy. Think about cows. For you, if you don't have food, you cannot feed on grass and you survive. Even if hunger strikes like right now, no human being can feed on grass and survive. But for cows, that's their delicacy. That's their favorite meal. Like Anita be like, I need some chicken. Some KFC. And that's her favorite meal. Just guess it. For cows, their KFC is they go to grass. They go to a grassland and fetch on grass. But for you cannot, even if you are hungry like cow, you can't fetch on grass. Why? Because grass is just cellulose. I don't know the percentage. I was going to quote, but I'm not sure. But it's just cellulose. Okay? And you don't have these enzymes cellulase to digest the cellulose. So you cannot feed on grass. Actually, when you eat food containing cellulose, the so-called dietary fibers, the refugees, the same way you've eaten that food, the cellulose will be passed out in poo as cellulose. But it's good, eh? You read about functions, importances of dietary fibers or refugees. It is good, eh? But it won't be digested. Why? Because you don't have this enzyme. Similarly, cows and other herbivores don't have the enzyme. They don't produce the enzyme cellulase. Now somebody is saying, "Yee, then how come they are able to digest?" Yes, they don't produce the enzyme cellulase, but they do form what we call symbiotic relationships with bacteria, with a given species of bacteria that this enzyme cellulase. And now this enzyme that has been produced, it will be the one that will digest the cellulose into glucose. And then the glucose will be absorbed and taken to the respiring tissues for respiration. And then you see your cow. gaining some butts, developing some chicks, and be like, yeah, my cow has a tupu. Okay? Yes, that's brilliant work. That's brilliant work. So uses of cellulose. Number one, we can use cellulose to produce rayon, which we can use to manufacture tire cords. The tire cords are here. These ones. So this is tire and this is rubber. The whole of this is rubber, which is vulcanized, whatever. This is rubber. But inside, to strengthen the rubber, then we add their tire cords. And these tire cords are made out of the cellulose. So you can see how plants are good. You can see how cellulose is good. Number two, the cotton, which we said it's 90% cellulose. We use it to manufacture clothes. You can see, you can have a look. Cotton, I think everyone at least has a cotton cloth. At least everyone here, I'm sure, among your clothes you have their cotton. Okay? You have their cotton. And cotton, it's 90% cellulose. It's what we purify to make cloth. And then some fibers. Then we have cellophane. Now this one is produced from cellulose. We can use it to package materials. I think I have here. So these are cellulose. This is cellulose cellophane. We use it in wrapping, in packaging, and stuffs like that. So that is cellulose. Then paper, I think, yeah. Paper. Actually, you people, if you're tired of exams, if you're tired of writing exams on paper, anyway, let me quote social media test. Somebody was like, guys, it's high time we became sensitive to environment. We should stop doing exams so that we can conserve trees. That was a social media post.
Amen.
Somebody is saying amen. So we should become sensitive to environment. Now, if you boycott exams due to paper, then we shall start setting online exams. And those ones can squeeze. If you want, I can give you by the way. There are
Huh?
apps which you can download and you prepare an exam. Then a student just comes in to do. It switches on a camera, it monitors whatever you're doing with your desktop. And then you just do. You focus your eyes on the camera. I'll be watching from here. So if you boycott exams because of paper, then we shall introduce that. But anyway, all papers, they are made out of cellulose. It is the one which is purified, and you make paper. Then I'm sure people here are young. You never saw this. You never saw cameras with such. People, all of you here, I'm sure you are 2000 kids. Some of you are 2006, 5, 7 there. Anta, how old are you?
Eh?
Uh-huh.
I don't know.
You can imagine. But you might be some 2008, 9 there. So you don't know this. Now, but for us, our ages, we used to have cameras with these photographic films. Okay? Yes. So these films, they can be derived from cell. However, there is a lot of science up to when you reach this, but you can have cellulose as a starting material. Okay? Yeah. So those of the 2000 kids here, I'm sorry you don't know this. You never saw this. But if you check your pictures, actually, those pictures which you are taken when you were young, they used this. But you were just too young. No. Then kiting, depending on your kindergarten, some people say cheating, others say kiting, others say kiting. So depending on your kindergarten. But it's cheating. It is also a carbohydrate that is made out of beta glucose molecules. So the groups are linked by a 1,4-glycosidic bond, a beta 1,4-glycosidic bond. In other words, it is identical to cellulose. However, the difference is that for it possesses this acetyl group, okay, that is attached. Actually, it is an acetylamide group. Something is missing here. Acetylamide group. I'll add it on carbon number 2. It replaces the OH on carbon number 2 on beta glucose molecule to form that acetylamide group. And because of this NH, so you can have a look. Anyway, you can have a look. So this is our cellulose. So on carbon number 2, here, instead of having the OH, you replace the OH with that acetylamide group. Okay? Even the same thing, acetylamide group. And now because it is formed from beta glucose molecule, the second one do somersaults, do rotates at 180 degrees Celsius. So that's why the amide group, our acetylamide group, is up. So it is also adjacent. Are you seeing? So it alternates. It is also alternating. You have it here, you have it here, you have it here, you have it here. Why? Because they are formed from beta glucose molecules. Now, because of this NH, this NH, when now the two adjacent chitin groups or chitin groups, I'm trying to accommodate people from different kindergartens. So when this NH, okay, of this strand, it's like this, approaches the NH of another strand, this is your N, and this is the H. You remember from organic chemistry, where we say that also nitrogen forms hydrogen bonding. So this one will be partially negative and this one partially positive, stuff like that. Partially negative and partially positive. So when they approach, then they have that dipole moment, where the negative will attract positive and that positive attracts negative. So these cross linkages, they will be formed between the two. These are hydrogen bonds. They will be formed between two adjacent groups. Okay? And so as a result, it will also end up having that parallel structure. Okay? That resembles the structure of cellulose. However, now, unlike cellulose, which is in plant cell walls, for chitin, you find it in insects, fungi, and some crustaceans. Okay? And some crustaceans. But it is also responsible for the formation of cell walls. You can have a look at this. You can have a look. You can have a glimpse. Yeah. Yeah, this is brilliant. Now, unfortunately, I can't take. So now, if this is my chitin, oh, now I've covered everything. But anyway, we can still see. So this is going to be our chitin, this one. So they are the ones which will arrange parallel to each other. You can see this is one strand, another strand, another strand, another strand. Okay, you can have a glimpse. And then these strands, they will arrange to form small microfibers, microfibrils. Okay, and these fibers, they will also arrange themselves, arrange to form large fibers. And the same applies, you can see when you try to magnify here. Okay, they will also be arranged into other small fibers. Then when we magnify here, okay, this is what we shall see until when we form our cuticle. So these, they will arrange themselves, okay, into strands to form an epicuticle, then an exo, and then the endo. Epi means above, upper. So this is the epi, then the exo, and the endocuticle. But the arrangement is kinda similar. And now it is this cuticle that will form the skeleton. Okay, you can have a look. So it is that cuticle that will form the exoskeleton in insects. Now in this cuticle we shall add their wax, and wax is a lipid. Okay, it is a lipid to prevent water loss in the epicuticle, especially to prevent water loss. And that's why the insects, they are good organisms as far as maintaining water is concerned. But cuticle chitin, it is made up of simply a carbohydrate. And which carbohydrate? This one, chitin. Any queries? You can ask again. Pardon?
What does the glucosamine ring mean?
The glucosamine ring mean. Glucosamine, it is a glucose with a short chain of with a short peptide chain. Okay, with a short peptide chain, e.g. in peptidoglycan. We are going to find it. Probably in our next lesson, because today's time is up. We shall find peptidoglycan, but you can do some reading. So the process of adding a short carbohydrate chain on the protein is called glucosylation. Okay, and you remember in cell biology where we talked about, is it glycoproteins? Yes. So the process of formation of these glycoproteins is glycosylation. So when you add a short peptide chain on the glucose, on a long chain of glucose, e.g. peptidoglycan, or a short carbohydrate chain in a protein, that is glycosylation. So a glycosylated compound, a glycosylated peptide is that protein with a glucose molecule. So I hope that one is okay. Maybe we can talk about inulin, because that one is just simple, and then we call it a day. So with inulin, this one is just a polymer of fructose molecules. Simply, when fructose molecules add up together, they will form inulin. Okay? And this one is a storage carbohydrate. It's a storage compound in most plants. In some plants, e.g., the Jerusalem artichoke. This one. I don't know if we have these plants in Uganda. The red onions, this kind of carrot, and we have these onions in Uganda. So they are storage compounds. The inulin is a chain of, you can mute. It's okay, you can mute. So the inulin, it is a carbohydrate chain. It is a chain made up of fructose fibers, or fructose molecules, not fructose fibers, fructose molecules. And it is for storage, e.g., in such plants. And you can have a look at the structure. Okay, so you know this, you have the OH here, and you have the OH here, and you have the OH here. So there are those OH groups that do condense, okay, together to form those bonds, the glycosidic bonds that will hold the fructose molecules together. So I think we shall call it a day. We can meet in our
Teacher,
next lesson. Yes?
teacher, can there exist beta and alpha fructose?
No, I don't think. I do not think. If there is beta and alpha fructose, fructose is fructose. Maybe if you continue and study deep, but I've never encountered one. Okay?